Hey guys, my name is Max and welcome to Simple Biology. In this video we're going to go over some of these biomolecules that we've been talking about in this unit and just try and identify them so that you can have that skill, which will be very useful for your AP tests and for any biology class that you take. So here's our first biomolecule that we're going to go over. I'm going to give you a couple seconds to kind of figure it out on your own, and then I'll get into it. Alright, so the first thing you're going to want to notice with this biomolecule is that it has a lot of oxygen. You can see there's an oxygen double bond to this carbon, and there is these oxygen and hydrogen hydroxyl groups on each of these carbons down the middle. Of course, at each of these junctions between the lines, at each intersection, there's also a carbon there. So this is actually going to be a 1, 2, 3, 4, 5, 6 carbon biomolecule. And the reason I point out these all these oxygens is because when these have all these oxygens, you can usually assume that's going to be a carbohydrate. And so this will actually be a carbohydrate. And from here, we can go into kind of the more specific type of carbohydrate that it is. And like I said, it's a 6-carbon sugar, and they're all covalently bound together into just one subunit. So this is actually just the monomer, which is called a monosaccharide. So yeah, this is a monosaccharide, which is a carbohydrate. All right, this is the next biomolecule we have. Go ahead and try and solve it on your own. All right, the first thing that should jump out to you with this type of biomolecule is that you have these 4 rings. You have a ring here, a ring here, a ring here, and finally a ring here. Whenever you have these four rings, you can basically assume it's a steroid. So 4 rings. Oops, sorry. 4 rings. It's a steroid. And so I kind of work backwards on these ones actually, and because you know it's a steroid, it's also a lipid. And lipids, of course, are any they're characterized as a nonpolar hydrophobic biomolecule, and like with our previous diagram, each of these junctions, each of these turning points in this case, are carbons in the diagram. And because there's all these carbons and there's also hydrogens bonded to these carbons, this is going to be a very nonpolar biomolecule. There's only 2 oxygens, and so there's just a very low amount of polar bonds. So this is a lipid, and it's a steroid because of those 4 rings. All right, here's our next biomolecule. Again, like the last 2, go ahead and try and solve it on your own before I explain the answer. Okay, this example is very similar to that first example, and the first thing you want to notice is all of the oxygens that are just kind of scattered throughout the molecule. And the characteristic thing about this particular biomolecule is that many of the oxygens are found in these hydroxyl groups, where it's an OH bound to the carbon chain. And when you see all these oxygens, the first thing you're going to want to think about are carbohydrates. Carbohydrates have a lot of oxygen. And so they'll actually have a 2-to-1 ratio of hydrogen to oxygen, which if you go through and count, this biomolecule actually does have that 2-to-1 ratio. So this biomolecule is a carbohydrate. Now the next thing you want to notice is that there are 2 of these like hexagon-shaped rings, and so I'm just going to show you the separation between the 2. And because of that, you can see that there are 2 subunits, and so this is something that we'll call a dimer. In this case, it's a disaccharide. Remember that a monosaccharide was the monomer of carbohydrates, so a disaccharide would be a dimer of carbohydrates. So it'll be 2 of those monosaccharides put together. All right, this is our next biomolecule. Again, go ahead and try to solve this before I give you the answer. Okay, this biomolecule, I'm just going to go ahead and say it is actually a lipid. And so the way I figure that out is because of all the carbon and hydrogen. Again, by looking at this diagram here, you see that you have all these carbons. Each carbon, there's a carbon at each of these points that it bends at. And when it says nothing, that means that all the rest of the carbon's bonds are with hydrogen. So that's carbon and hydrogen. We call this a hydrocarbon tail for this particular molecule. And because of all that carbon and hydrogen, which are nonpolar, it's going to be a nonpolar molecule, which is a lipid. Now, you see that there are three tails, and this is a very specific kind of biomolecule that we talk a lot about. And when you have these three tails, they're made up of what are called fatty acids. Each of these tail represents a fatty acid, along with the carbon and oxygen bonded to that tail. And then this other part of the molecule is what's known as glycerol. And so when you have all three of these fatty acids bonded to a glycerol, you have what's known as a triacyl glyceride or a triglyceride. So I'm going to go ahead and write triglyceride. It might be helpful to know both the names, but triglyceride is easier to write, so that's why I'm writing it. Now again, yeah, so finally the last thing we're going to do to classify this is you see that there's this double bond right here. And because of this double bond between two carbons, this fatty acid chain, or this fatty acid tail or chain, is actually bent at that particular location. So it's got a kink in the tail. And so because of that that double bond, we call it saturated, or we call it unsaturated. And the reason it's unsaturated is because you could potentially have two more hydrogens attached to this tail, but it has less than the total amount it could possibly have. So this is an unsaturated fat or triglyceride, which is a type of lipid. Here's our next biomolecule. Again, try and do this on your own before I give you the answer. All right, this biomolecule is incredibly similar to the last biomolecule, and so you probably got it already, but I'm just going to write down what we already remember from the last time. It's a lipid because, again, the number of carbon and hydrogen in these tails, number of carbons and hydrogens, and it's a triglyceride, just like the last one. And you can tell this because of the three chains. Tri means three, so there's three of these chains, and this is a glycerol molecule here, whereas the glyceride comes from. Now notice that unlike the other one, there are no double bonds in these carbon-hydrogen regions. There's no double bonds between two carbons. There are double bonds here, but we don't count those. We only count double bonds between two carbons. So this is a saturated fat or saturated triglyceride, and it has as many hydrogens as it could possibly have for that number of carbons. So again, this is a saturated triglyceride, which is a type of lipid. All right, here's our next example of a biomolecule. And again, like this last, all the other ones we've done in this video, go ahead and try and solve it first before we get into how we're going to name this biomolecule. All right, you'll probably notice that this biomolecule is very similar to the last two examples we gave because it has this same zigzag line, the same hydrocarbon chain, and it also has this group over here that we've been calling a glycerol group. It's still got that same little subpart of it. And so it's actually going to be classified as a lipid just like the other two. Now you'll notice that there is still a little, there's a little charged region over here and here. This whole side group here is going to be polar, but we're not going to be looking at that quite yet. For now, you can see that these fatty acid portions make it a lipid. So it's a lipid, and just like the last one, it's a saturated lipid, so I'm going to write that down, saturated. But now we're going to address this last little problem, this side group here. Now you can see right here there's a phosphorus molecule, and because this phosphorus has 4 oxygens on either side, it's called a phosphate group. So that phosphate group is going to be very important for naming this biomolecule. It's actually called a phospholipid. So any lipid that's going to have that phosphate group in place of one of the fatty acids is called a phospholipid. You'll also see that there's this little extra piece attached to the other end of the phosphate. Some phospholipids have that, some don't. It depends on the particular kind, but don't let that get you confused. Because it has that phosphate, it's called a phospholipid. All right, here's another example. Again, go ahead and try and solve it on your own before we give you the answer. All right, like some of the other examples that we've given, the first thing you're going to want to look at is the oxygens that are just kind of scattered throughout this molecule. They're really just everywhere, and I'm circling them all right now so you can see them. But because it has so many oxygens, we know that this is a carbohydrate. Remember, carbohydrates have much more oxygen than any of the other groups. Again, also if you look, you can see that there's the 2-to-1 ratio of hydrogen to oxygen. If you add the hydrogens that are attached to all these carbons on the joints between the lines, and so that 2-to-1 ratio also tells us that's a carbohydrate. Now, the next thing we're going to try to look at is it a monomer, is it a dimer, is it a polymer? And you can see that they have these nice little subunits, this hexagon shape, and there are actually 4 of them in this example, but this little squiggly line tells us that it actually goes on more. So because it has more than just one of those subunits, we're going to call it a polymer. And in this example, for a carbohydrate, we call that a polysaccharide. So this is a polysaccharide, which is a type of carbohydrate. All right, here's our next example. Again, go ahead and try and solve it on your own first. All right, this example is actually pretty different from anything we've seen so far in this video, but kind of the first thing you should notice is that there are these carbon-nitrogen bonds here. I'm just going to kind of put a line where each of them is. Oh, sorry, that was the wrong side. It's actually these carbon-nitrogen bonds, where you have a carbon with an oxygen and then bonded to a nitrogen. So anytime you have these three atoms here in a chain, you're going to want to definitely notice that. And what you can kind of see is that there are these very important carbons, every couple of carbons on this chain, where they'll have a nitrogen on one side with some hydrogens, a carbon and an oxygen on the other side. In this case, it's a carboxyl group. And then a hydrogen, sorry, there's actually supposed to be a hydrogen here too. And here, they'll have a hydrogen on one side, and then what's known as the R group. I'm going to box in the R group. So this R group here is basically just the thing that's different for each of these subunits. And because of just how I've been describing it, you probably have realized by now that each of these is called an amino acid. And so whenever you have this, this group I boxed in, this is going to be called a protein or a polypeptide. So I'm actually going to call it a protein. And like I said, each of these subunits between where I broke off those bonds is called an amino acid. Make sure you know that terminology because that'll show up as you go through your biology courses. So it's a protein, and it could also actually be called a tripeptide because it has three of those amino acids. But for our purposes, it's a protein, or you might even call it a polypeptide. I'll write that to polypeptide. All right, here's our last example. Again, go ahead and try and solve it on your own before I give you the answer. Okay, this is actually, in my opinion, one of the most distinctive of all the biomolecules that we have in this unit, because it has 3 different parts that are all very, like it's hard to really get it confused with the other groups. Sorry, my pen's not working very well. So I just kind of circled those 3 groups, and so of course you have a phosphate group here. You have some sort of sugar, some sort of ring structure here, and then this other thing that has these nitrogens on it. So because it has that nitrogen, you know it can't be a lipid, it can't be a carbohydrate. You don't see any of the normal things that you'll see with a protein. You don't have that nitrogen and the oxygen and the carbon on next to each other. So it's actually a nucleic acid. And nucleic acids will always have this phosphate group here next to a sugar, in this case ribose, and then that'll also be bonded to the nitrogenous base. And so these 3 groups are what make up any nucleic acid. And because there's only one of each, there's only one of this subunit, we also call it a nucleotide. That's the monomer of a nucleic acid. So this is a nucleotide. If you had more of these groups all bonded together, you'd have a backbone of the phosphate and of the ribose, or in the case of DNA, of deoxyribose. You'd have those two as a backbone. It would just be alternating, and so you see a bunch of these kind of piled on top of each other to see if there would be a whole chain for nucleic acid. But in this example, there's only one of each. There's just the three little subparts, and so this is a nucleotide, which is a type of nucleic acid. Thanks for watching. I hope that made a lot of sense, and if it didn't, then go ahead and see the quiz that's attached to this video for more practice, because it's really just as simple as that.